Welcome to Click for IB Biology. This playlist concerns neural signaling, and here we're going to look at how the resting potential recorded within the axon of a nerve cell is established, and then we'll actually go on to look at a comparison of impulse velocities in different types of nerve. There's a blank version of my presentation. That's a PDF. You'll find that in the description below, and there you'll also find a link to a 10-question quiz with answers that will allow you to consolidate your understanding of this part of the course. So the resting membrane potential is a charge which is found across the cell membrane, and you can see it here in this image of the axon, and it measures at minus 70 millivolts, and this is recorded on an oscilloscope in millivolts with a reference electrode in the outside extracellular fluid here, and the recording electrode is within the axon plasm, the cytoplasm of the axon, as you can see in this simple image here. And when this is performed, experimenters have found that minus 70 millivolts is sustained over time. This is time on the axis of the graph here. At minus 70 millivolts is sustained, and this is achieved by two parts. Part one is something familiar to you from cell membrane structure, which are the sodium-potassium pumps, and they initiate an exchange of sodium ions in a ratio of 3 sodium ions are pumped out of the cell with an exchange of 2 potassiums coming into the cell, and overall this initiates a negative charge within the axon. But this is amplified by a second protein in the membrane, which is called the potassium leakage channel. We need to call that a channel, not to be confused with the potassium gates which we studied in cell membrane. This is a different one. And so this is the second part, which is then able to bring the resting potential to minus 70 millivolts within the axon plasm as recorded on the oscilloscope. So there are two-part explanation there of the charge. Now, when we observe nerve cells, whether they have Schwann cells or not, we can measure the speed of conduction of the impulse, the velocity of the impulse, if you like, along the axon, which is shown here. Now, the nature of this impulse is actually a wave of sodium ions, high concentration sodium ions, moving along the axon, and how it achieves that is the subject of the next couple of videos in this particular playlist. But let's just take a quick look at the speed of conduction. Where we have no Schwann cells, we have non-myelinated nerves, and their conduction tends to be around 10 meters per second. Now, this is characteristic of invertebrate organisms, but we also, as vertebrates, possess some of these in parts of our nervous system, in sensory nerves where we experience dull pain, and in other parts of the grey matter of the central nervous system. So it's not exclusive to invertebrates. We do have some unmyelinated nerve axons as well, and so they're slow conductors of a nerve impulse. But with the insulation of the axon by the Schwann cells, this accelerates the conduction with what's called myelination to approximately 100 meters per second, so much faster. So myelinated is faster than non-myelinated, and so we find these in motor nerves in particular. Other relationships to velocities which can be found is that if we take the diameter of the cross-section of an axon and we plot that on the x-axis, and we measure the speed of conduction down those nerve cells. So down here we've got small diameters, up here we have larger diameters. Then we find that there's a high correlation coefficient between these two variables, so a strong relationship. That's what a correlation is. It's a relationship, and designated, as you can see here, as R. So that R value tends to be quite high, around about 0.8. A typical organism within vertebrates, the way that they increase their speed of nerve conduction is to have large diameter nerves, and the most famous one of these is found in the Squid giant axon, which was used for much of the initial research on nerve conduction, which has a very, very high rate of conduction because its diameter is large. Now, the data for this, plotted around this line here in order to generate the regression line, of course, the data fits very, very well with coefficients of determination, capital R squared. Note that it's capital R squared, and this coefficient of determination is round about 0.8 to 0.9, meaning that 80 to 90% of the variance in the data can be accounted for by this relationship. In other words, 80 to 90% of the data fits this trend line that we've been seeing here. And these two types of relationship evaluation, the correlation coefficient and the coefficient of determination, may well be used in your internal assessment and the statistical analysis of data. And to our final relationship here is that if we look at the size of the organism, then we tend to find that the speed of conduction of the nerve impulse decreases. Now, when you measure the actual velocity in axons, the speed has not actually significantly reduced. They're still high in this area here. The issues actually arrive with the complexity of the connections between the central nervous system and the peripheral nervous system, and the types of connections which are used along those large limbs, and the distance from which the impulse has to travel. So distance becomes an important feature in this conduction. It's not really the speed, but more the time to conduct which increases. Okay, so now what we're going to do is we've established what the resting potential is. Now we're going to work out how the impulse is actually begun. There's a chance to subscribe to the channel, always appreciated, of course, and the playlist is here for your convenience, so you can quickly check out the other videos which have to be studied in this part of the course.